All right, I think we'll get started. Welcome to the CFAR Monthly Seminar Series. Our speaker today is John Sleasman. Before we get started, I just want to again remind everyone, please make sure that you're muted. And we'd like to handle questions either through the chat feature or wait till the end of John's talk, and we can then handle any questions then. So again, thank you all for attending. John came to Duke, as many of you know, in 2013, following an illustrious career at the University of Florida and then University of South Florida. He is currently the Glenn A. Kaiser and Altha Muriel Kaiser Professor of Pediatrics and Chief of the Division of Allergy, Immunology, and Pulmonary Medicine in the Department of Pediatrics. His research and clinical expertise focuses on primary and secondary immune deficiency diseases, as well as immunodiagnostics. John currently serves on the Executive Scientific Leadership Committee of the International Maternal Pediatric Adolescent ACTG. He was the former chairman of the NIH AIDS Immunology and Pathogenesis Study Section and chaired the Therapeutics Leadership Group for the Adolescent Trials Network sponsored by NICHD. John's CV lists over 140 publications related to both basic immunology and clinical research, and includes studies to elucidate the mechanisms of acquired and primary immune deficiency disease. He is currently studying the complexity of chronic macrophage activation to understand how HIV infection results in inflammation and its implications in both cardiovascular and CNS morbidity among youth living with HIV. And John has enjoyed over 30 years of continuous NIH support and continues, I think, in his collaborative efforts with Maureen Goodenow, who many of us know who is the director of OAR. So John, welcome. It's been too long since we heard from you a long time ago. So we're all anxious to hear updates on what you've been up to. So take it over, John.
Great. It's really appreciate the invitation, Ken. It's great to see you guys, if nothing else, by virtual communication. I think that the real theme for this talk is going to be around how do we optimize the use of our clinical trial networks in studying basic mechanisms of HIV pathogenesis, and more importantly, when life deals you lemons, how do you make lemonade? So I'm going to talk about our NIDA-funded project on marijuana and inflammatory pathways in youth with HIV. So I had to update my slides. The CDC website is only good through 2018, and the depressing news that this shows is that the epidemic in terms of new infections still is in mostly young individuals in the Southeast from underrepresented minorities, with the 25 to 34-year-old group being the highest, and then the 13 to 24-year-old being down here as well. And this is a group that's very hard to reach, as I'll show you in the next slide. And in aggregate, if you look at the under 30 age group, that makes up about 40% of the new infections in the US. What's even more problematic is that these youth tend not to be engaged in care, with only about half of them being diagnosed in the 13 to 24-year-old age group, and then a very large proportion of them not being virally suppressed. So this is the epicenter of our epidemic, is right here in these two age groups. And as that slide showed, things have been pretty flat. We're not making much improvement. So for those of you that are old like Kent and me, you remember that when the epidemic started in the 80s and early 90s, we had a huge pediatric HIV epidemic as it became obvious that the virus could be transmitted from mother to child. And NICHD and NIAID formed the AIDS clinical trial groups to address this. And one of the most important studies that came out of that was ACTG 076, which showed that the use of zygovidine would block maternal transmission from mother to child. For those of you that are younger and writing grants now, all of a sudden, my grants went from being highly significant to statements from the reviewers like pediatric AIDS is going away. Why do we need to study this? NICHD, in its wisdom, decided to pivot and provide funding to study the HIV epidemic in adolescents. And they were the first group to start looking at this in a systematic way. And with funding from NIAID and NIMH, they formed the Adolescent Trials Network back in 2002, which really was the HIV, it was the Adolescent Trials Network for HIV/AIDS interventions for prevention of HIV in youth. And I had a leadership role in two protocols that we're going to talk today. One was a treatment protocol called ATN 061, which was to look at preservations of immune subsets in youth who received antiretroviral therapy. And another one, which Christina might be interested in, was ATN 071101, which is an assessment of inflammatory markers associated with neurocognitive impairment in HIV-infected adolescents. So we've had biorepositories associated with these two studies. Those biorepositories live in MSRB1. And we have all of the clinical data on every single youth that was enrolled in these studies available from how far they got in school to their shoe size to just a very, very rich database on youth. One of the things that came out from our early studies of youth who were relatively newly infected, and this was news at the time, but it's old hat now, was that there was this persistent macrophage activation with persistently high levels of soluble CD14, even in those youth who had achieved full viral suppression, and that this soluble CD14 remained elevated despite optimal suppression of viral replication with ART. And one of the nice things about these studies that I'll point out here is that the ATN also was wise enough to have a cohort of HIV, of youth without HIV to serve as controls. That was part of their interventional studies to prevent transmission, so that every site also had a balanced demographic of youth without HIV that were part of the clinical studies. And that's where this HIV negative youth came from. So we were interested in looking at this in a little bit more detailed way within this cohort, and we wanted to look at the inflammatory biomarkers that really differentiated youth with HIV from youth without HIV. And we did a very complex study where we looked at associations between a multiplicity of pro-inflammatory biomarkers for lymphocyte, macrophage, and vascular inflammation. And as you can see from these red lines, which represent the associations between the youth with HIV versus the controls without HIV, which is notated in blue, and then the shared pathways, there's a lot of concordance or associations. For example, a soluble CD14 was associated with also elevations in IL-10. And there were associations between these markers of vascular inflammation like SVCAM, CRP, and ICAM, the soluble CD163. So we're starting to put together a kind of a matrix of how these pro-inflammatory cytokines might interact in the pathogenesis of these youth that were undergoing early treatment. Through a PCA analysis, we were able to see that HIV-infected youth, regardless of whether they were treated or not, because some of these kids were untreated. This was before they went on ART. Some of them were treated but failed and had viral loads of greater than 50, and some of them were treated and were fully suppressed with viral loads less than 50. You can see that they had relatively overlapping on this diagram, and that the factors that most discriminated HIV infected from the uninfected group were really inflammatory markers associated with inflammation. Soluble CD27, which is, as you know, a member of the TNF family, soluble CD14, which is through the toll-like receptors, soluble CD163, TNF alpha, and LPS. What was interesting to us, and this is different from, John, you like to study older adults, You'll remember that IL-6 is a very important pro-inflammatory cytokine in older individuals, but this was not discriminating in our youth. So there are some subtle differences between our population of young folks with HIV as opposed to individuals who have been living with the virus for longer periods of time. We've been studying the inflammatory consequences of HIV-infected youth in terms of bone, cardiovascular, neurocognitive, and metabolic, and have published a number of studies. We were the first ones to show this idea of low bone mass in behaviorally infected, HIV-infected young men. It was interesting that we only found this in men. We did not see this in the women that were enrolled in these cohorts. We were able to show that macrophage activation played a major contribution to the bone loss by sex among these youth. We found out that tenofovir was not the culprit, and that the tenofovir in and of itself had minimal effect on the biomarkers of bone health among youth. And then we were also the first to identify some of the early markers of endothelial inflammation with the identification of SVCAM as an early biomarker of cardiovascular disease among the youth enrolled in these studies. So I'm going to switch gears for just a minute instead of bragging about all the clinical studies we've done and talk a little bit about how we got into the monocyte macrophage business. Back early on at Florida, we were studying monocyte-derived macrophages and macrophage-tropic viruses. Maureen took the major role in doing this. And since I was an immunologist, I said, well, maybe toll-like receptors play an important role in macrophage activation by HIV. So Joe Brown, one of the graduate students working in the lab, took it on himself to do some beautiful studies on this, published a number of papers, but this one from PLOS in 2008 was probably the best received. And what Joe was able to show was that HIV infection has a big effect on macrophage signaling pathways like cytokine pathways through JAK-STAT, apoptotic pathways through FAST, the MAP kinase pathways, and calcium pathways with the p-values, into the negative stratosphere in all of these. But toll-like receptors will seem to be independent. That is, there was really no effect and no difference in toll-like receptor signaling when we infected monocyte-derived macrophages with an M-trophic virus ex vivo. So he said, sorry, John, you're wrong. What we do know, for example, from our clinical studies that microbial translocation induces activation of monocyte macrophages through LPS binding through TLR4 and soluble CD14 to induce both the release of CD14 as well as activation of pro-inflammatory cytokines when in the presence of the virus. And this leads to a lot of these comorbidities associated with HIV. So what we know from a summary of the macrophage activation studies is that monocytes and macrophages are primed for activation through new pathways mediated by both LPS and HIV, that chronic macrophage activation results in persistent inflammation, and that macrophage activation is not reversed by antiretroviral therapy. At the same time we were doing those studies, there was a huge interest in this. I think that one of the most important pathogenesis papers that published in probably the last 10 to 15 years was from the SMART study that showed that soluble CD14 was the best predictor of overall mortality in HIV-infected adults. Dana Gabusta and her group were able to show that high levels of LPS and soluble CD14 correlated with the extent of HIV encephalopathy, and that HIV-infected adults with elevated soluble CD14 had a 3 to 4 higher risk for atherosclerotic disease compared to adults who have the similar risk factors. So we took advantage of another study that we did in the youth. Now, the premise of this study was also wrong. The premise of this study is if you started antiretroviral therapy early, soon after diagnosis of HIV infection in newly identified youth, you would be able to improve their neurocognitive function. So this was a 3-year study of 128 youth that were followed for 3 years who started ART when their CD4 counts were quite normal. You see that their the CD4 count at the time of entry was 658. They were on antiretroviral therapy for majority of them for over 144 weeks, and about 60% of them had viral loads below the level of detection. One of the things that we learned was that it didn't really alter their neurocognitive function over the 3 years that we followed them. There was really no measurable improvement in their function. But what we did learn that was a little bit surprising to us was the high rate of substance use among this group, and it was primarily tobacco and cannabis. 39% of them were using cannabis almost every other day. And tobacco use in combination with cannabis was extremely common, as I'll show you as well. We've also studied depression, sort of a sidelight. We've studied depression and depressive symptoms as it relates to neurocognitive function in this youth, and we were just able to publish a paper looking at the depressive scales, which takes us off in a whole different direction, but I just wanted to point that out, that we have the whole Beck's depression inventory and very good data on depression in this population as well. So the average length of HIV infection in these youth at the time that they entered on the study was only about 10 months, and we were able to follow them for 3 years. What we found in terms of the neurocognitive testing was that verbal memory and visual spatial memory were most impaired. I wish I would have guessed attention and executive functioning would have been most impaired, but it was really verbal memory and visual spatial memory. There was little change, as I mentioned before, there was little change in these domains over time. And motor and attention and executive function was relatively spared in the youth that we followed. So to remind you guys that don't think about the brain all the time, visual and visual spatial memory maps discrete areas of the brain, primarily in the spatial memory, cortex memory, and most importantly, the hippocampus. And if you remember from your neurobiology, the hippocampus is the highest rate of glial cells. There's a lot of impact of inflammation could potentially have a role in neurocognitive functioning in this particular domain. So we wanted to study whether or not our biomarkers of inflammation had any impact on visual spatial memory. And this was a study that was done in collaboration with our folks at UNC. So Kevin Donovan and Matt Loop are over at UNC. Julie, Bernie, and Bill are here at Duke. So really this is a, even though this is an ATN study, this is really a Duke UNC collaborative work to examine the relationship between these inflammatory biomarkers and neurocognitive performance in the youth enrolled in ATN 071. What was really interesting, and I think that the guys over at the coordinating center and the statistical core that Mike Hudgens runs over at UNC did a spectacular job on this project, because it was complex. Like I said, we collected a lot of data on this, and what Kevin and Matt were able to do was to look at the biomarker alone and then do adjustment, which controlled for age, gender, race, socioeconomic status, substance use, depressive scores, and concurrent infections, and then finally bringing into the model adjustments for covariance, including viral load and CD4 T cell counts. And no matter what we did, no matter how we made those adjustments, visual spatial memory was most impaired within this population, which to me really confirms that there's some meat to this idea that HIV infection in youth really has some very discrete effects on neurocognitive function, particularly in this visual spatial area. We did see some other things that, like executive function. It was interesting that the biomarkers correlated, but then when we did the adjustments, that correlation was lost. So chronic macrophage activation and neurocognitive function. So elevated plasma soluble CD14 levels were associated with impaired visual spatial memory in our youth with HIV. There's now a lot of evidence that soluble CD14 is associated with microstructural white matter and damage to other neuroinflammatory states in a variety of things, from Alzheimer's disease to CTE to multiple sclerosis. TLR is expressed in the microglia and probably plays a role in neurodegenerative disease. And LPS induced TLR activation in hippocampus and pyramidal neuron nucleus may have some effect here. And I think there are some models that would prove that in animal systems. And we should really think about therapeutic interventions that target macrophages as a way to improve visual spatial memory in youth with HIV. So that's all published. And now we get to the good part, okay? So I'm going to talk about the HIV as a model to study the anti-inflammatory effects of cannabis. And this is really what we're doing with our current U01. So I did not know this until I got to Duke, but you have to have a sexy title for your clinical trials. I just called it the NIDA Project. And Lisa Haito said, no, can't do that. You have to have a sexy title. So we had a competition, and Alyssa Barberi, who works for Lisa Haito, was the winner. And I'm not exactly sure how this worked, but she came up with Synapse and this nice little logo with the blunt here, the neurons, for studying youth to determine nicotine and marijuana's role in immune activation and neuropsychology while looking at substance abuse effects in youth with HIV, AKA Synapse. Now, remember, I mentioned to you that when you have, when life gives you lemons, make lemonade. So we put the substance profile, we used a WHO tool called ASSIST to do self-reported substance use in all the youth that were enrolled in the protocol. And we also did toxicology, and we were surprised how truthful our participants were in terms of reporting their substance use. And we had our youth without HIV and our youth with HIV populations. And only about a third of them did not use any substance at all. And this would confirm again with the toxicology. Marijuana use by itself was only in about 16% of our youth with HIV and only about 7% in our youth without HIV. But marijuana in combination of tobacco was the combination of choice. Now, this is an interesting thing that I learned from this study. We asked the question, do you smoke cigarettes? And the answer was no. But we then asked the question, do you use blunts? John, you know what a blunt is? You obviously are not a follower of the Rastafarian movement. They prefer to take their marijuana, to take the edge off it, is to roll it with tobacco and smoke the two together. And that's called a blunt. And this was the preferred route. So 40% of our youth are actually about 30% were using the blunt, because only about 9% of them were using cigarettes, tobacco alone. So most of them, and the blunt was the preferred way to use this. So given that the fact that we had a lot of marijuana use, I had to learn a lot about how CB1 and CB2 works, and we include Tom Klein, who is an expert in cannabis, as a collaborator on this project. I've gotten to kind of get a little bit comfortable, not real comfortable, in terms of understanding the pathogenesis of marijuana on pro-inflammatory cytokines, and it's not simple. Dave Murdoch helped was the driver behind trying to put this together. But for those of you that follow this, cannabinoids, of which there are many, THC and CBD get all the press, but there's probably over 100 different phytocannabinoids that have effect on G protein coupled receptor signaling in humans. But CB1 and CB2 are the primary ones. CB1, as you know, is the one that gets you high and expressed primarily in the brain. But CB2 is highly expressed on T cells, monocytes, macrophages, B cells, and NK cells. And unfortunately, we found from some of our early studies, the expression of these receptors is very species specific. So studying them in rats and mice does not necessarily correlate with the signaling that we see in humans. The other thing that's important to remember is that these decoupled protein receptors influence a lot of very important cell signaling pathways, including cell cycle, cellular proliferation, cell death, angiogenesis, and other pro-inflammatory pathways as well. So we started out by doing going back to doing some ex vivo experiments with monocyte-derived macrophages to see if we could measure these going back to that Joe Brown study that I talked about, where we take monocyte macrophages and stimulate them in the presence of interferon, and then looked at signaling. We wanted to see whether interferon primed macrophages would respond to how they would respond to THC ex vivo. So in these studies, what you can see is the presence or absence. Vehicle means no THC, T means THC. At different micromolar concentration, you can see this dose-dependent decline in the production of this inducible chemokine CXCL10. So this gave us some evidence that there was some effect by THC on interferon-induced macrophage priming. So we took this data, and then we went back into our population of youth that were enrolled in ATN 071, but this time we looked at them by substance group. So we used a reference group from our youth without HIV who did not use substance, and this was our reference group of 25 individuals. We had a group of youth who were unaffected, who were heavy users of alcohol, marijuana, and tobacco. So this was, we thought, would be a control for the reference group in terms of substance use exposure. And then within our HIV, our youth with HIV, we had youth who did not use substance that had detectable viral loads of greater than 50. And then we had 3 groups of individuals who were fully suppressed over the 3 years, who had undetectable viral loads, who did not use substance, used marijuana alone, or used marijuana in combination with tobacco. The groups were mostly male. They were all quite young. The ages were between 20 and 24. These are slightly older, or slightly younger, because they were recruited at the same time that the study started. So we had a blood draw at the time of study, and then we did the blood draw at the end of the study, the 3-year study, so that that's why they're about 3 years older. Mostly African American. They had been on antiretroviral therapy for a long time. Even among those youth who failed, they had relatively low viral loads, and their CD4 numbers were robust all the way across all the groups. So we looked at the global blood transcriptome. This was work that was done in Maureen's lab and continues to be done with her lab up in Bethesda. And then we looked at differentially expressed genes and perturbed pathways within the group. And we also looked at substance use as it related to chemokine gene and protein expression. I think it's important that when you start to look at these studies, because there's more and more of them coming out, that we really be careful in terms of how we define our populations. We just can't mix them together. We were very rigorous in terms of how we defined our populations, and I'm glad we did. As you can see, there's a difference between the youth and the unaffected youth and youth that are suppressed in terms of plasma CXCL10 and CXCL9 levels. But there's also a big effect that these chemokines normalize when viral replication is taken under control. So we really don't see as much of a robust difference between groups, and other groups have published this, but they haven't been quite as rigorous about how they define the substance use profile, or whether or not viral suppression was total. And I think this is going to be important in interpreting these studies in the future. What we did learn for both of these chemokines is that there was a strong association between the level of gene expression and the level of protein that we could measure in the plasma. And they held up pretty robustly, particularly for this CXCL10, which, if you remember, was the ex vivo study that I showed you earlier. We also looked at differentially expressed genes, again, with using the youth who were uninfected and did not use substance as a reference group for our fold change. And again, you can see that in the youth that are unsuppressed, there's a number of genes that are up or down regulated within the chemokine receptors and their ligands, ranging for as high as a threefold change for CXCL10. But what you can also see is that when viral replication has gotten under control, a lot of these genes then normalize. But there are a few that remain elevated. And what was really interesting to us, for example, for CXCL9, as well as CCL5, and for those of you that remember, CCL5 is RANTES, the gene normalized in the gene expression normalized in the group that used marijuana alone. That's the group 4. The other thing that was really striking to us in looking at these is that even though this group 5 that used marijuana in combination or tobacco are fully suppressed, just like group 4, we can start to see these perturbations, these differentially expressed genes in some of these same pathways that were dysregulated in group 2, particularly like CCL5, CXCL9, and CXCL10 are now, their expression has gone back, is now going back up again. When we looked at the DEG and gene pathway analysis among youth by substance profiles, another one of the striking things that we saw was that group of youth who were not infected but used substances, there was no differentially expressed genes in this population. There was no impact of substance use on gene expression in this group. But when you look at inflammation in the context of HIV infection, whether they are unsuppressed or suppressed, you start to see some real changes in within the transcriptome, with over 3,000 genes dysregulated in our group 2 and 1,100 genes dysregulated in the group that used marijuana plus tobacco in the backdrop of suppressed infection. We then looked at some of using these IPA analysis. We looked at some of the canonical pathways to see whether they were perturbed. This is a kind of a complicated way of looking at pathways. So these are pathways that are already defined and the genes are known. So we're not looking at a biochemical pathway here. What we're looking is a genetic expression, and then looking at Z-scores for differentially expressed genes in order at a mathematical formula to determine whether a pathway is up or down regulated. And what you can see, obviously, is there are many, many more pathways perturbed in the youth who do not use substance but have uncontrolled viral replication. But within the youth that are suppressed, there are 12 pathways that we made perturbed. Some of them actually shared among particularly with the group that use tobacco plus marijuana with the unsuppressed group. So we disaggregated the genes within these pathways to look at the effect of marijuana alone or in combination with tobacco on gene expression. So again, this is our a group of unsuppressed youth with no substance, and you can see there a number of unique genes, EEGs, that are unique to the unsuppressed group, but also a number of genes that are shared with the marijuana plus tobacco group that are not shared with suppressed marijuana or the suppressed no substance group. There's also a series of very unique genes here that were unique to the marijuana only group, including genes, for example, like KRAS and FAD that are associated with cellular proliferation and with apoptotic pathways. And so we're starting to get these unique profiles of how substance affect the transcriptome based on the viral, our ability to control viral replication. So to summarize, cannabis suppresses CXCL10 production by interferon-induced macrophages ex vivo, and that marijuana can normalize chemokine gene expression in vivo in viral suppressed youth on antiretroviral therapy. The effect is subtle in terms of the plasma effects, but CXCL10 and CXCL9 gene expression does correlate with chemokine, and we're exploring this further now in this project. Recreational use of marijuana had little effect on the gene transcriptome in uninfected youth. So everybody that's standing in line to buy CBD oil may not be getting the effect that they hope to get. But I do think that in the context of inflammatory, underlying inflammatory state, there may be some potential benefit to use of cannabis and cannabis-like substances as for anti-inflammatory purposes. However, recreational marijuana and marijuana used in combination with tobacco perturbs multiple genes and multiple canonical signaling, and marijuana alone or when used in combination results in substance profiles that involve key immune and key signaling pathway. And that, as I mentioned a minute ago, marijuana when used tobacco results in perturbation of inflammatory pathways associated with ongoing viral replication, that there may be some clear deleterious effects of using marijuana in combination with tobacco in fully suppressed individuals. Recreational marijuana use alone or in combination with tobacco causes measurable effects on the inflammatory pathways the use are suppressed. And the gene based on the gene expression that we've looked at, marijuana may be predominantly anti-inflammatory, but in contrast, marijuana with tobacco is maybe pro-inflammatory. And ultimately, I think this is a really important area for us as HIV investigators to really look at it in a little bit more detail. There are no trials going on right now looking at cannabis. There is one in Canada that's ongoing right now to look at the CBD effect on inflammation in individuals living with HIV, but there's nothing going on in the US, and there's a number of reasons for that. And so I think I've intentionally left time for discussion. So I want to acknowledge the folks here, Julie, Bill, Bernie, and Joan Wilson, who has helped us with this project. Over at UNC, Matt Loop and Kevin Donovan from the ATN Coordinating Center, Christina DeParis, who has been helping us figure out some of the pathway analysis and inflammatory pathways specifically in this group. And Lisa Hightow, who's been absolutely fantastic. She is an incredible individual in terms of understanding the epidemic in youth, and she is she's going to be a real leader in this area in the future. We have a collaborative site at the University of South Florida through Christina Rodriguez and Sharwoot Sembawit, who are actively recruiting, and these are individuals who, as the CFAR starts to think about expanding our collaboration, these are very good sites in the state of Florida with good clinical trial infrastructure. Sharon Nichols at the University of California, San Diego is a neuropsychologist who's been helping us do all of the neurocognitive studies and has been working with us now for over 10 years. And then Maureen's group at the Office of AIDS Research, Kai Finn, Lee, Sam Ashok, and then Julie Williams and Sophia Appelberg worked with Maureen at the University of Florida as postdocs before she left to go to OAR. So I said I was supposed to be done in 45 minutes. I did it.
Nicely done.
Yes. So.
Very good, John. Let's open this up for questions. I mean,
yeah.
Just that was a real tour de force. I just have a generic question. How much of this that you've done in the youthful population is informing what may also be going on in some of the more adult and elderly patients with HIV?
Ken, I don't care. So thank you for asking, though. It's really interesting. And I'd really like John to weigh in on this. There's a paper from Rizzo that looks at this in, this is a group at Michigan State. They've been looking at substance use among their cohort in Michigan. Their results are somewhat similar, particularly looking at things like macrophage activation and CXCL10, but their effects are much larger. And I think it's because they're, first of all, they have a much larger population. They didn't segregate their groups out by smoking and non-smokers. So when I say smokers, I'm talking about tobacco use. So a lot of their observations are sort of colored by the overlay between the use of cannabis plus the use of tobacco. The other one is that their overall levels of pro-inflammatory cytokines were much higher than our youth. They're not real rigorous about how they define viral load. That's really the best group in the US that are looking at this. Now, Nicholas Tremont and some of the guys up in Canada have embarked on this study, and they have a nice a paper that they published in the British BMJ. I think that's British Medical Journal, right? With some of their preliminary results, but they're really more focused on T cell activation than they are macrophage activation. So this is, I think, a unique niche for us if we want to pursue it. We have the capacity to study it in the youth, and I think it would be interesting to look at this across the lifespan to see whether, what the similarities or differences are based on age as well as length of infection.
Yeah. Okay, let's open it to other questions. Please, if you have a question, just show yourself and state your question, please.
I don't see anything coming up in the chat yet, so anybody
no. John, while we're waiting, let me just pursue that a little bit further. Again, I'm under the impression, and there's not a lot of data on this, that in older individuals with HIV that are now stably suppressed, the amount of their, if you will, immune reconstitution while on virus suppression is really dependent upon what their CD4 nadir was when they began their suppression. Do you think that macrophage activation is tied in there also, or do you think that the macrophage activation you're talking about is completely independent of what's happening with T cells.
Well, based on what we're seeing in, and this is mostly data from the 061 study. So the 061 study, we started ARV when this protocol first initiated was back in the days when you didn't start ART until CD4 counts declined below 350.
Yeah.
And we, this was one of the first studies to actually to push early treatment. And what we wanted to see was the extent that we could preserve many of those subpopulations. We did some very extensive slow looking at naive T cells, effector memory cells, and T cell function in this group. And we were able to show you we did get improvement in that population. The two things that came out from that study that we published, I guess probably 2014, 2013, I can't remember exactly. It was one, is that there was still some evidence of T cell activation, even in those youths who had been suppressed for 3 years, compared to the uninfected group. It was mostly in the expression of 38DR on effector memory cells in that population, downregulation of CD28, but it did not correlate with the soluble CD14.
Okay.
So we didn't see that. Now, soluble CD163 came down a little bit at about year 3, so macrophage activation did start to wane, but the question that you're asking is how much does macrophage activation contribute to ongoing T cell activation? I think that's still something that is a little gray.
Okay. All right, we have a question from Christina Mead. Oh, she needs to hop off. Sorry, she's excusing herself.
We have an Elvis sighting here. Wilton's on.
Christina, thank you. Others.
Hi, John. How are you? Kent, can I ask a question? This is Wilton.
Absolutely. Wilton, go right ahead.
So, John, this is a B cell question. So, I'm curious to know if the inflammatory state in these individuals suggest any or provide any signatures that would suggest an impact on B cell responses.
Absolutely, yeah. As you know, Lee published a paper in Frontiers on this while you were still a graduate student in Maureen's lab. It is the ongoing effect of HIV replication on B cell activation, I think is real. If you remember it in that study, we compared patients with lupus and patients with HIV and saw very similarities in B cell dysregulation in those groups with loss of overall B cell diversity. And I think for those of you that are working on vaccines and think about this a lot more than I do, I think that's something to keep keep in mind. Also remember that B cells are much more like macrophages in terms of their activation pathways than they are T cells. So they express class 2, they can serve as antigen presenting cells. And this is an area that people have danced around, but nobody's really taken that on directly to really look at where the B cell abnormalities are and how it affects the repertoire, I think is another really important question that needs to be asked. Bart asked me about 15 years ago about, for those of you that don't know, I'm a rheumatologist too, about CDR3 length in lupus, and do lupus patients get HIV? And for a long time, Bart was going around the country saying that lupus was relatively protective against HIV, but I think that's been disproven since then because of the CDR3 length. But I think it's a really important question.
Thanks, John.
Thanks, Wilton. Other questions? John, I've got a dozen of them, but you mentioned during your talk that we should be focusing on targeting macrophages. How do you go about doing that?
Well, most of the trials from probiotics to statins have not been particularly helpful in lowering macrophage activation. You know, I think there's probably been at least 6, if not 7, clinical trials through the ACTG targeting, you know, Michael Letterman's been leading a number of those to try to look at, you know, targeting specifically at TLR4, but also to lower many of these pro-inflammatory cytokines that are associated with the comorbidities. I'm not too enthusiastic about most of them, but I do think that based on some of the data that we're showing here, we could make an argument for use of the cannabinoid receptors as a potential therapeutic intervention. I'm really curious to see what comes out of this Canadian study that's taking place right now.
Yeah, good. Well, we have a shy group of people here, not.
So I will emphasize that we have infrastructure here, and we are open to collaborations. I have been trying to drag Dave Murdoch kicking and screaming into the world of youth, trying to get him to reimagine himself as a young man. But I
am a young man.
Try to think back, Dave, instead of
I'm with
forward, right? but I do think that the ATN is going to be recompeted soon. I predict that Lisa will be a key player in that, and with the close proximity and the ongoing collaborations that we've had with the UNC group, this would be a great area for us to kind of synergize. The other thing, and I'm sorry that Christina had to leave, the other thing for us to really think about it, this is a great interface between behavioral science and immunology. That's something that we tend to be on parallel paths rather than converging paths. But the fact that we were able to identify with pretty good reproducibility, a link between a neurocognitive domain and a specific activation pathway in monocyte macrophages, I think are really important. We're starting to look at this now in some of the depressive symptoms as well. So I think that that's another area that our colleagues in behavioral sciences and those of us that think about lymphocytes, macrophages, B cells can start to interact more as well.
Yeah, John, I'm glad you brought up the biorepository, because that could be a tremendous resource. Is there a mechanism for approaching you with concepts?
Yeah, you have to walk into MSRB1, walk down the hall, turn to the left, and look for Birdie. Yeah. I can't take this stuff with me. I think one of the problems with biorepositories is we tend to sit on them for decades and never really use them. And so my dream is that when Dr. Klotman finally throws me out of my ear, there will be nothing left in that ATN repository, that we will have used it among investigators, and we have plasma, we have cells, but most importantly, we've got this very robust clinical data on these kids over a long period of time. They're just not snapshots. They're 3 years worth of data, if not more, in terms of, because we really went to a lot of trouble to capture this.
It would be great. Mary Orris brings up the issue: could CFAR share information on the repository? Is there something we could help with? I mean, I could even think about, we have a developmental core, and we have some occasionally have targeted RFAs. I mean, we could
Yeah.
even think about a CFAR targeted
also, what I'm really thinking about, Kent, because those studies are now starting to get old. I mean, we finished ATN 071 in 2014. We need to kind of boost this up. And what I've been thinking about in terms of the recompetition for ATN or even the renewal for this grant is to try to start to build cohorts and biorepositories in the new in these new clinical trials as they go forward in youth. We don't have a youth biorepository at all, either in the IMPACT or in the ACTG. If you look at what's available in the ACTG biorepository, it's mostly from individuals, with a median age in the mid 40s. But to have something that has that, that 20 to 30 age group with a large population, would be really valuable because they're underrepresented in the ACTG biorepositories. Other people are starting to get interested in this, particularly in light of the push to end the epidemic, the PASHA initiative. We have a little bit of a head start, and certainly with the connection with the adolescents, we can do that. I don't know whether you remember Karina Rodriguez here is an ATN site from when I was at the University of South Florida, and she's got a very robust population. So those are groups that we could bring on board to help contribute to our CFAR biorepository as well.
Okay.
We
ought to chat more.
That stuff.
Yeah, we ought to chat more and see how CFAR can help facilitate some of this. And again, whether you know it or not, you're part of the CFAR internal advisory committee that we're looking to convene sometime.
Yeah, I still have the letter. I think the letter's right here.
Yeah. Well, again, COVID kind of put a damper on that, but again, we hope to do that meeting. And again, I think that would be a good time to think about how CFAR could help facilitate some of this, because again, it is a tremendous resource, and we'd love to see it actually continue and grow. And so let's put our heads together in the future and see what we can do. This is an exciting area that I think you've identified here.